the fossil of the RNA would that we're still we don't have any protein as such which is able to synthesize protein. We still have to depend on RNA.
RNA rules. We all know.
Yes. That's a fair statement.
No, I'm going to pass. I'm going to pass in the interest of time. Yeah.
Okay. Well, good.
Okay, well, let's thank Matthew. All right, so it's my very great pleasure to end this session by introducing Tom Rajbandari. Someone asked me why we call him Tom instead of Uttam. I don't know, but that's what he told us to call him. From the last two talks, the Elegance last two talks, you can see his great influence on science. And as we've all commented, Tom is so modest, he never really talks about his own accomplishments, but they've been extraordinary, and he's been really a central part of the Khorana Rajbandari legacy. Because it's Tom, I'm going to let you talk as long as you want. So here we go.
Let me see. Where is the thing? Okay. Okay. Great. Yeah. Thank you, Marcia. I needed that. This morning, Dieter Sol, who I have known for close to 60 years, reminded me that I'm the oldest person here at this meeting. Yeah. He's only 6 months or 4 months younger than me. So, I went to, when I joined Govind's lab, it was October 1, 1962. I think it was a Friday, and arrangement was that I would go visit Govind on Monday so we could talk about what I was going to do, because he wouldn't let me know beforehand what I was going to do. And on Saturday or Sunday, I went out desperately looking for a place to get something to eat. And it was some kind of a small drugstore that also sold food, and the person sitting at the place was Dieter Sol. So he had also just arrived maybe a month before, whatever. Yeah. And the interesting thing is that was not the first time I met Dieter. I had met him at the place where I did my PhD, and so Dieter was spending a month, not month, a year, because all German PhDs have to spend a year in another institution, and he was spending the year at the same lab where I was doing my PhD. I was going to do my PhD. Yeah. So that's how I know Dieter. Right. So I met Govind, and he told me what I was going to work on. He was going to give me a project that he himself was working on, because he was going to move on to better things like the genetic code. This was 1962, right? So that what I got from Govind was that I would work on some way of putting tags on the ends of nucleic acids. Nothing was known about nucleic acid sequence, nothing. Some way of putting tags on the ends of nucleic acid sequence such that we could begin to get some information in much the same way as Fred Sanger put tags on ends of proteins or peptides and with DNP and started getting sequence information of peptides. So basically same principle. So he handed me his notebook. And I looked at the notebook. When I went home, I looked at the notebook, and it frightened me. It frightened me because Govind is a meticulous note keeper. He wrote, he did an experiment, right? And he put the time, right? And what the results were. And so one day's notes would read like 6:30 p.m. Finished this, put samples on the counter to count, went home to eat dinner. And then it would start with 8:30 p.m. same night. Yeah. And then he described whatever he did until about 11:30. And then the morning would begin. So something like that. So that really frightened me, because it basically told me what the expectations would be, without actually telling me that I was expected to do that. So that's really, but it was a wonderful thing, of course. opportunity. And so this is what I was doing, basically. He had, people talked about coenzyme A yesterday, how John Moffat synthesized coenzyme A. John Moffat synthesized coenzyme A by using something called phosphoromorpholidate, right? So what I was supposed to make this compound here, methyl phosphoromorpholidate, and react it with that phosphate here, and that way put a tag, that with C14 label tag. And then if that reaction worked, and then I would isolate that, and then I would cut it at this site and isolate the radioactively labeled nucleotide and identify the nucleotide. So this was the idea. So I did that. After a while, I was succeeded. I did that on a total tRNA of the cell. So it was a mixture of tRNAs. The idea was nothing wasn't known. So really was, I was interested in doing that. So and it was known that there was a lot of G there at the end, which is what I found. But when I did a two-dimensional chromatography, I found there were 5 spots. 4 of them were in A, C, U, and G as expected, and a 5th one which remained unidentified until later that turned out to be pseudouridine. So this was the beginning of my work at Wisconsin. And then we talked about what to do next. Well, even before we published the paper, then we talked about what to do next. And it was after about a year after I arrived there, I think. So we said, no, I think people are getting interested in tRNAs because tRNAs are obviously very important. So the idea was now that we would now start purifying tRNAs and actually see if we could sequence a tRNA. So that was my job. And Govind was incredibly generous to me. He gave me 3 postdocs and 2 technicians, and I had a lab downstairs, right? And so 3 postdocs, 2 technicians, and just left me alone, literally, except to go upstairs and go to the seminars and do things like that. So that was, and then as I was trying to sequence tRNAs, paper came out from Bob Hawley's lab, who had actually sequenced the tRNA. 1965, he got a Nobel Prize for that. I shared it with Govind. Right. So anyway, we continued with it because so many tRNAs, and that was not the end of the world, So we were working on a different tRNA, and we sequenced the tRNA, and that turned out to be the phenylalanine tRNA of yeast. So this was a very, very useful tRNA, really, because people really started using this tRNA for all kinds of reactions, all kinds of purposes. It was the first tRNA that was crystallized by Alex Rich and in Cambridge at MIT, and by Aaron Cook at the MRC labs in Cambridge, in England. And so that was very useful thing. But this tRNA was interesting. It shows it's actually 76 nucleotides long, and it contains 14 different modified bases. And this is really a lot of modified bases. And with this tRNA, then we could begin to do experiments now, biological experiments. And the first, and again, the Govind's lab had a lot of collaboration going on between people. And the person I would collaborate, for first collaborative experiment I did was tizosol. So what was happening was, so this tRNA is interesting, right? The sequence, it has the anticodon sequence GAA, right? And Francis Crick in 1966, we sequenced this tRNA in 1966. We reported partial sequence at the Cold Spring Harbor, published this paper in 1967. And then Francis Crick had meanwhile published some idea called wobble hypothesis, which said that you do not need a perfect pairing between the messenger RNA codon and the anticodon of this tRNA. There is some wobble. Wobble means some, you can have more than just perfect hydrogen bonds. That if you have a G here, that G in the anticodon can pair with either U or C, and therefore read 2 different codons for the same amino acid. So that was just a hypothesis. And we here with this purified tRNA now, we could do, and protein synthesis experiments going on in Govind's lab, and Dieter's expertise in it, we could now do the experiment, right? And so we did the experiment, which actually proved that Crick was right, at least in that particular prediction. Okay, so this is the experiment where we now took that tRNA, we attached the radioactive amino acid phenylalanine to that purified tRNA, right? And then we added it to a protein synthesis system, and we asked, does it read both the poly UUU and UUC codons on in the translation system? And you can see that the radioactivity present in the tRNA goes into proteins polyphenylalanine with both poly-U and poly-UUC. So this single tRNA can read both the UUU and the UUC codons, the first proof of wobble hypothesis. So that was one collaboration. And then we were collaborating with others in the process of doing a completely, innocent experiment. Really, it's just a control experiment. We came across an enzyme which is now called peptidyl tRNA hydrolase. And that paper we published with Hans Kossel, who was another guy in the lab, doing something completely different. So that was this experiment here, where you attach a radioactive amino acid to a tRNA and modify that radioactive amino acid. The amino group is modified with an acetyl group, so that you have acetyl amino acid attached to the tRNA, or you can have a short peptide attached to the tRNA, and then you treat it with a cell extract. All right? And what you see is that all of a sudden the radioactivity just goes. So there's an enzyme in the cell extract that is removing that acetyl amino acid, right? And then we know that the tRNA is left intact because it can now be charged with amino acyl tRNA synthetase. So the amino acid can be put back in. So this was really the discovery of peptidyl tRNA hydrolase that Umesh continues to use still. So this was another really useful collaboration. And then we became interested in initiation of protein synthesis. And at that time, there was a paper that came out from Cambridge in England, which said that the initiating tRNA in E. coli is the formyl methionine tRNA. It's the tRNA that's charged with methionine and then formulated. Umesh mentioned that before, right? And so the question was, what happens in eukaryotes? So that was interesting. So we thought what I would do is we would just see if we can take a yeast tRNA and see whether there's any yeast tRNA that can be amino acylated with methionine and be formulated, thinking that maybe that would be also the tRNA used for initiation in the eukaryotic system. Right? So that was the idea, and we did that. And indeed, if we take a yeast tRNA, by that time we also fractionated yeast tRNAs into two types of methionine tRNAs. And if we took those tRNAs and added amino acid, and then added radioactive formyl tetrahydrofolate, formyl group radioactively labeled, what you see is that with one yeast tRNA species, the formyl group goes in. With the other yeast tRNA species, the methionine tRNA species, it does not. One methionine tRNA, there is no formyl group incorporated. The other methionine tRNA, there is formyl group incorporated. So that said that indeed in yeast also there is a tRNA which can be actually amino acylated with methionine by using an E. coli enzyme, and then the E. coli enzyme will also then add a formyl group to it. So to us, this meant that maybe this is the tRNA that is used for initiation of protein synthesis in eukaryotes, about which nothing was known then. All right? So we became interested in it, and I think at that time we started doing protein sequencing the tRNA. In those days, each sequence of each tRNA took about 2 years. Literally 2 years, a lot of effort. That's why 3 postdocs and 2 technicians. And then we sequenced it, and then I moved to MIT in Cambridge, and then the first thing we did, first paper I published from there, was a paper in collaboration with Harvey Lodish, done by one of his students, who is now a very well-known genetics researcher, also a faculty member in our department. And we showed that this tRNA, indeed, which is the so-called this formulatable yeast tRNA, the putative initiator tRNA, was actually initiating protein synthesis in yeast, which said that in eukaryotes also protein synthesis is initiated with a special methionine tRNA, but not, it turns out, with formyl methionine. You do not have to formulate it, as we all know now. So this was really the beginning. And then this, the tRNAs became something that we really, just very interested in. Important things to do with protein RNA recognition processes and things like that. And I think for the rest of my life, I have stayed literally very, very close to the tRNA field. Things we got sidetracked on working with mitochondria for about 15 years. Matthew mentioned that, right? So, all right. So now, what I'm going to tell you today is really something simple and it's nothing complicated, and just something that we've done in the last few years, and that has to do with basically the biology of tRNA in archaea. Now, archaea is, as you know, there are three kingdoms: bacteria, eukaryotes, and archaea. And this archaea is what we will focus on. And I'm going to really talk about two different aspects of archaea. One is how the genetic code is read in archaea, and the second one is why there are no nonsense suppressors that have been found in archaea. You might be, it's just not something that people know. Is that in bacteria, nonsense suppressors play such a critical role in genetics of bacteria and viruses. In eukaryotes also, nonsense suppressors have played a very critical role in genetics, in yeasts and worms. And yet, in this third kingdom, archaea, nobody has come across any nonsense suppressors ever. The question is, why not? So those are really two things I would like to talk about. All right? So, new insights into reading of isoleucine codons in bacteria and archaea. Why is this interesting? This is the genetic code. Now, this is a little different from Dieter. Dieter has given you the very latest version of the genetic code. A lot of his very beautiful work. Right here, I'm showing you the old genetic code. Right, this is the genetic code that is, to me, is very beautiful. Yeah, looks so well organized. It consists of 16 4-codon boxes, 64 codons, right? And it is you actually it belongs to two sets. One set you have all one set you have all 4 codons specifying the same amino acid in yellow, right? And the other set specifying, so there are 8 of those 4 codon boxes, and then the other set specifying where you have each 4 codon box is split into two. The 2 with pyrimidines specifying one amino acid, and 2 with purines specifying a different amino acid. And there's 6 such boxes. So the 14 codon boxes then are like that. And then the 15th one is here. 2 of them again specifying one amino acid, but the other 2, one is a stop signal and the other one is a tryptophan signal. So that's also, okay, so now this is, right, so according to wobble hypothesis, a single tRNA with G in the anticodon can read any codons ending in C or U, and a single tRNA with U in anticodon can read codons ending with A or G. Right, so if we go by that rule, then we can explain basically how all of these codons can be read by sets of tRNAs, but without any ambiguity in the process. But here, with this system here, this one exception here is that you have a 4 codon box split into 3 and 1. Now, you can imagine that these 2 codons would be read by a tRNA, and this codon would be read by a U. But if that does, what prevents it from also reading that G? And if it does read that G, then it's a mistake, and that cannot be allowed. So there has to be a system here, right, that somehow there's a system that allows you to read the 3 codons for an isoleucine without also one of the tRNAs also misreading the AUG, which is a methionine codon. Now, there is some information on that. In bacteria, it is known that you have the U and C is read by a tRNA that has G in the anticodon, and the A in the tRNA is read by a compound that is a modified C, a C to which a lysine is attached, right? And that's called lysidine because lysine is attached to it. Exactly how that happens, how that reads a C, how that reads an A specifically without also mispairing with G and reading AUG is not known. Okay, so we did some work. We decided now what is the story with archaea, and we have found that in archaea also, it's the C, modified C, but the modification is different. You have something called agmatidine. We call that thing agmatidine because it's in bacteria you have lysine attached to it. In archaea you have agmatine. Agmatine is a decarboxyarginine attached to it. So, okay, so okay, so this is it. Now, all right. So this is the story here now. With in both cases, you have one tRNA that reads AUU, AUC for isoleucine, and here a second tRNA with a modified C that reads A without also reading AUG. Okay, and so the question really is, how does C base pair with A? It's counterintuitive, right? How does C base pair with A and not with G? Obviously, it's modified, and so this modification is really playing an important role. So to really get some idea of what's going on, I talked to Venki. Venki and I have talked over the years for years and years and years. So we collaborated with Venki Ramakrishnan, and we sent the samples to his lab. And the idea was that we would do a crystal structure analysis, have the tRNA bound to the ribosome, and see how the base pairing is going on between the modified C and A. Right? Now, there were some ideas in the literature that one way that a modified C could base pair with A is that the modified C exists in different types of tautomeric structures, and the tautomeric structures were these two. You can imagine this looks like a U, right? And similarly, this looks like a U. So you can imagine that these two tautomers would actually base pair with A without base pairing with G, right? And so the question is that correct? So I will just show you here, this is agmatidine, and this is lysidine. Lysine, and this is agmatidine, right? Okay, so the answer came from Venki's lab after some effort. This is the system that is used. It's you have the ribosome, you have a very simple message, AUA, right? And then you have your tRNA that we have sent him. And analysis, the results were very surprising. The surprising result was that, first of all, there's only a single hydrogen bond linking C to the A. So this is it, a single hydrogen bond linking C to the A. And really perplexing, how can that happen, right? And secondly, answer was that there's another hydrogen bond between the long agmatine side chain. There's a hydrogen bond donor and hydrogen bond to the phosphate of the messenger RNA. So you have that is anchoring that there, and just a single hydrogen bond that is enough for the C to recognize an A. So that looked good. It still did not explain why C does not base pair with G. And then the answers came. I don't think this tells you much, but let's go here. That if you now try to build a model with G there, what happens is there's a serious steric clash between this long side chain and the side chain of G. A group called G. So that is really what it makes, preserving its specificity for A without allowing it to base pair with G. So that was, to us, was a reasonably satisfying answer, and that's where things stand. Okay. All right, so I think that's really good enough. Now, so then we asked, okay, we got answer, right? But why, why do we have a system that uses a modified C to base pair with A and not a U, and just put some restrictions in it so that U can base pair with A easily? So the thing was we wanted to now see if we mutate the C of the tRNA to a U, what happens? Can we isolate the tRNA and see what its base pairing potential is, right? And we did that. And this is just show that it's purified and all that stuff. And we do all the analysis. And the answer was that indeed the tRNA, the wild type tRNA with agmatidine base pairs exclusively with A, but not with any of the other three isoleucine codons, right? But all with AUG. But the mutant tRNA with a U here still base pairs very well with AUA, but it has the potential for mispairing with AUG. What that is in fact suggests that indeed, if you put U there, you have the potential for misincorporations of things. So that is where it stood, and we also characterized the U that was there, and it turns out to be also modified. And modified U is here. This is analysis. These are the 4 markers, and the compound that we found is that. And that compound happens to be something called cyanomethyl U. They're not important, but but it's kind of explained to us why nature has not used a U at that site and used this bizarre way of modifying a cytidine, right, to base pair specifically with A without allowing it to base pair with G. All right, so that's number 1. So I'll go on to Marcia. You have to tell me when I need to quit. All right, okay, okay, all right, great, okay, I'm glad. Yeah. Okay, so nonsense suppressor and archaea. Yeah, I mentioned that suppressors have played such a critical role in bacteria and eukaryotic genetics, and if we could have suppressors in archaea, maybe that would also facilitate archaeal genetics. And so that was the reason why we asked why are there no suppressors in archaea. And the question we asked is, well, can we just express a suppressor tRNA in archaea and see whether it will work in suppressor? Whether it's just something that suppressors just don't work, or suppressors are inherently very toxic to archaea and things like all those are possibilities. So people in the lab took 2 different tRNAs, mutated the anticodon in such a way that you could get amber, ochre, or opal suppressors. They would read UAG, UAA, and UGA codons, right? From serine and tyrosine, just in case one is a strong suppressor and one is not, and all that stuff. So just to make sure that we get some positive results, basically. And so you put that into the cells along with a reporter system that allows you to see what's happening here. And indeed, this is just this expression system. I don't think we're interested in it. We can either have it a constitutively expressed tRNA or inducible system to express it, just in case the tRNA really happens to be toxic. And indeed, for this now beta-galactosidase is being used as a reporter. And you can see that if we put the beta-galactosidase with an amber mutation in there and also express an amber suppressor tRNA, you get suppressor. So it says that there's nothing wrong with archaea. Archaea will actually allow suppressor tRNAs to function. And this is a plate assay here. And if we have put an ochre suppressor will also read the amber codon to a small extent. And this is again similar to the in what we know in bacteria, is that in bacteria the ochre suppressors also suppress amber codons, to some extent. Okay, so that's good. So with that then, we decided to see whether we could actually put these suppressors onto, these are now be expressed, just not from the chromosome, but from a plasmid things, right? So we decided to ask whether we could actually isolate stable strains carrying the suppressor tRNA genes, because if we can, then indeed they would be quite useful, right? So we have introduced them to the chromosome using the standard homologous recombination system. We introduced them into 2 sites, one at the so-called beta-gal locus and another one at another locus, to see whether we can somehow regulate the expression levels of these by putting it into different loci and things like that. And indeed what we can see, and this now is just an inducible system we're using, inducible by tryptophan inducible. So just adding tryptophan to the system, you can induce this expression. And here we are looking at the, Okay, right, good. So here we are looking at tRNA levels, northern blots, right, here. And we can see that depending on the tryptophan concentration, the levels of tRNA go up, right? And that's a good thing. And then if we use the highest level of the tryptophan, and then do a time course, you also go, there's time-dependent increase in TR. So this is a completely inducible system and all that. And with that, we are all in business, and we also showed that actually you can have, you can also correct or suppress oxytropic mutations in the